So in the afternoon session, we will now switch from sporadic tumors, somatic mutations, more to the germline mutations and the hereditary tumors. And I guess you are all aware of the fact that now, after about 2 decades since the discovery of the high-risk genes, let's say BRCA1-2 or the HMPCC genes, we are right now facing a second era for deciphering the remaining heritability. And therefore, I'm very pleased that we have the first speaker to introduce Doug Easton, who is one of the leading experts, actually, and brains to accomplish this goal. And Doug is professor of genetic epidemiology and director of the Center for Cancer Genetic Epidemiology at Cambridge. He coordinates the Breast Cancer Association Consortium, a collaborative group that has collected data from thousands, from about 200,000 individuals. He performs and heads several studies, and within this consortium, numerous new risk genes have already been discovered that all go into the improvement of the Boadicea risk calculation model, and you will present the latest data on that. Please, Doug.
Thanks very much, Rita. It's a great pleasure to be back here in Heidelberg again on a beautiful day. So I'm being asked to speak about how to predict breast cancer risk, and perhaps worth reminding ourselves why we want to do that. And because we have various ways that we can, methods that we can use to avoid breast cancer or deaths from breast cancer, some of them listed here. So we have prophylactic surgery, we have screening, we have chemo prevention, we have certain lifestyle changes, and all of them are to some extent effective, but they also have disadvantages. So prophylactic surgery is obviously very drastic, screening is not particularly effective, chemo prevention is somewhat effective but also has side effects, and lifestyle changes are difficult to implement. And so in principle, the prevention of breast cancer would be more effective if it could be targeted at those women who would really benefit from it, and that's really the basis of this, the idea of trying to identify high and low risk women. And we've been doing this for a number of years now, and most countries have some sort of method for triaging women into their risk level. These are just the guidelines that are issued by NICE in the UK, which triage women into so-called near population risk or moderate risk or high risk, and various modalities are targeted to people in these risk groups. So for example, women at high risk are the group who are proposed for MRI screening, for example, or chemo prevention. So and other countries, Germany included, have similar guidelines. So the idea of risk stratification has been around for quite a long time, but at the moment it's largely based on family history and BRCA1 and 2 status. And one of the issues is how to extend that in a more rational way to include other pieces of information. And I'm particularly going to concentrate on this high-risk group, which can be roughly defined as on the basis of the risk over the next 10 years being more than 8%, or a lifetime risk of more than 30%, or a relative risk of more than about 3-fold. So there are various factors you can bring into the question of who gets breast cancer. There's genetics, which I'm mainly going to talk about, but there's also lifestyle and reproductive factors that we've known about for a long time. And there's also things you can measure by imaging, particularly breast density. And all of these factors contribute to defining who's at high risk. And one of the main questions is how do you put all that together? Within the genetic space, it's also quite a complicated picture. So historically, we think of BRCA1 and BRCA2 because they're the most important high-risk genes. But there are 2 other major categories. There are common risk SNPs, of which we know a large number now. I'm mainly going to talk about that first. And then there are also a number of other so-called non-BRCA and BRCA2 breast cancer genes, of which we know much less about at the moment, but we're gradually coming to terms with them as targeted sequencing becomes more affordable. And I'll say a bit about those as we go through. So most of the data on the SNPs, as Rita alluded to in her introduction, comes from large-scale array genotyping studies. And this technology has enabled us to be able to study common variants throughout the genome on an affordable scale and hence on a large scale. And so there have been two really large studies which we've been coordinating. The ICOG study, which is about 100,000 individuals, also done for two other cancers, ovarian and prostate cancer, and the more recent OncoArray study, which is a somewhat bigger array and done on somewhat more individuals. And this has required the collaboration of something like 100 different studies worldwide from Europe, the US, Southeast Asia, Australia, and Africa. So a very wide collaboration of individuals needed to perform these studies. And together, it gives us a data set of something like 100,000 breast cancer cases and 100,000 controls. And you need a study on this scale because the effects of the variants individually are quite small. So you need a very large data set in order to get reliable estimates. And although the arrays themselves are slightly different, it's possible to combine them using a process of imputation, which essentially allows you to get estimates of the effect of any common variant in the genome. So we have data on something over 20 million SNPs in total. So when you put this together, you get some results like this. So this is these are so-called Manhattan plots. So each of these little circles is a SNP. The x-axis here is the genome, and the y-axis is the significance level. And you see these spikes. And some of these have been known for a long time. So you get this big spike here, which is close to the FGFR2 gene, which is the most important of the breast cancer hits. And then there's another one here, TOX3, there's another one here on 11q near CCND1, and so forth. Some of those have been known for some time, back for the early days of the GWAS. But if you take away what you already knew and then you look at what's left, you still see a large number of highly significant association peaks. And in total, if you put all the available together, there are about 150 of these. What do we know about them? I mean, each of these has a biological story behind them, and we're only just beginning to understand some of what's going on. Some of them are close to quite plausible driver genes. Some of them correspond to genes that are also important somatically. But in this talk, I'm not really going to talk about that at all. So we're just going to consider these as labels for individual loci. One thing we do know, if you divide, subtype is important here. So if you divide associations into ER positive and ER negative breast cancer, that turns out to be fundamentally important. So the majority of the SNPs are associated with ER positive disease with little or no effect for ER negative disease. You'll also notice that the effect sizes are individually quite small, with only a few exceptions. So most of the effect sizes are relative risks of about 1.1. Then there's another subset of SNPs which are predominantly associated with ER-negative disease. So for example, variants at the TERT lockers and the ESR1 lockers. And then there's a third subset which are associated with ER-positive and ER-negative disease to a roughly equal extent, so pointing to some completely different biology, for example, the PTL-HLH lockers. So subtype is important in terms of risk prediction. How do you put this information together in order to make it usable in risk prediction Well, clearly individual the SNPs on them by themselves are individually not that informative. So to make use of the information in risk prediction, you have to put the effects together. And you can do that because it turns out that the effects of individual SNPs are more or less independent. And that means that you can combine the effects together in a so-called risk score. And that's illustrated here. So what we have here is the number of risk alleles that an individual carries, both in the general population, in the controls, and in cases. And in this study, which is from the BCAC, so on average, people carry about 65 or 70 risk alleles. Nobody carries no risk alleles, nobody carries all of them. You get this normal distribution. And what you see here is that the distribution in the cases is shifted a bit to the right. And that's what you'd expect because these are risk alleles, so they're more common in cases. And the extent to which it's shifted to the right is a measure of how predictive SNPs are in predicting breast cancer risk. So more formally, we do this by combining the data together in a so-called polygenic risk score, which essentially sums the effect of the risk alleles weighted by their effect size. And now if we use this risk score to look at the absolute risk of breast cancer, you can show that indeed they are quite predictive. So this is some data which has been published this year in the JNCI, and this graph on the left here are absolute risks for breast cancer overall, according to the polygenic risk score divided into quintiles. So you can see here the women in the top 20% of the risk distribution have a risk of about twice that of the general population, and the risk in the bottom 20% is about half. So a significant difference in risk of some importance. This is for ER-positive disease, so as one would expect, the polygenic risk score is more predictive for ER-positive disease because most of the loci are ER-positive loci, somewhat less so for ER-negative disease, and that's something we need to improve. It works, the risk stratification using the PRS works both for women without a family history and for women with a family history. So they're more or less independent of family history, although the absolute effect is bigger in women with a family history because they start off with a bigger risk to begin with. So PRS is potentially, in a sense, more predictive of risk in women with a family history. Now then there's, as well as the common variants, which we can combine together in a PRS, there's another part to the genetic story, and that's that area of rare variants over in the left-hand side of the graph I showed earlier. And that's, we've known about those for some time, but they've become sort of more fashionable with the introduction of gene panel testing. So a number of companies have become involved in the area of using panels of genes in risk prediction for breast and many other cancers. Just a few of them are illustrated here. So this is a bit more of a murky area, and you can see if you look carefully at the lists of the genes on these panels that they're quite different. So there's some commonality, but some which are unique. And the issue of which of these genes and which of the variants are really associated with the risk certainly hasn't yet been fully resolved. In this review we did earlier this year, we tried to establish from the available data which genes were their variants really associated with breast cancer risk, and we came up with this list of 11 where we thought the evidence was fairly well established, which is certainly a lot less than's on most of the panels. You notice a couple of things here. Firstly, that the frequency of the variants is generally very low compared with the SNPs. And also you notice that this is an estimate of the relative risk associated with a truncating variant in these genes. And in most cases, it's very imprecise. And in some cases, it's almost impossible to get any sort of reliable estimate. So this is a bit of a grey area. I just highlighted one gene here, PALB2. So this is one gene where I think there's both good evidence for an association, and the risk is, if you like, in the high risk category. So that's a little bit, along with BRCA1 and BRCA2, a bit unique. The others, the risk are generally lower. Another thing to be aware about is that it's not to believe everything that you see in the literature. So this is an important gene which is on a lot of the panels, BRIP1, involved in the same DNA double-strand break repair pathway as BRCA1 and BRCA2 and PALB2. Variants in BRIP1 are clearly associated with ovarian cancer, but they're not, at least the available evidence suggests they're not at all associated with the breast cancer. So these panel tests need to be used with caution. And so this is sort of some of the data illustrated in terms of absolute risks here. This dotted line here, this is the 30% high risk threshold. So you can see that a truncating mutation in BRCA1 and BRCA2 and PALB2 exceeds that threshold. But for example, CHEK2 or ATM doesn't. So if you're going to consider these variants, you need to consider them in combination with the other risk factors. How predictive are the different risk factors in determining risk? Well, you can express that in a rather similar way to the way we express the effect of the SNPs, and that's to combine them together in a score. And the predictive value of the different factors you can express in terms of the width or the standard deviation of this distribution. And so here's some examples of the different factors and the corresponding standard deviation. So you've got lifestyle risk factors, breast density, SNPs, and the other genes. And it turns out the lifestyle risk factors have the standard deviations about 0.37. It's a bit similar to what you can achieve with breast density. Breast density is a bit better if you use a quantitative score like Cumulus rather than a categorical score. The SNPs are a bit better still. The other genes don't contribute that much, but where you really get a big effect is by combining it all together, because then effectively you get the combined effect of all these risk factors. And then you get the standard deviation of about 0.78. What does that mean in practice? So this is just another way of looking at the sort of same data. So this is in terms of an ROC curve. So the red line here is the ROC curve for all the SNPs taken together. That's a bit better than you can achieve with breast density or lifestyle risk factors. If you put it all together, you get this green curve, which is, again, a bit better. It gives you an error under the ROC curve of about 0.71. If you look at this in terms of absolute risk now, in terms of how discriminatory that would be, so the blue line here is, again, this is the distribution of the overall risk score in the general population. The red line is the corresponding distribution in cases, which is shifted to the right a bit more so now because we're combining all the risk factors together. So if you use this sort of curve, which is still a bit theoretical because we're making some assumptions about how you put these factors together, you can identify about 4% of the population with a risk of more than threefold, so high risk, and they account for about 18% of all breast cancer. At the other end of the spectrum, about 20% of the population are so-called low risk, so a risk of less than a third of that in the general population, and they account for only about 4% of breast cancer. So you could argue that these women should be taken out of screening altogether. And just by way of comparison, BRCA1 and 2 mutations are about 0.4% of women at high risk. So by putting all this information together, you're effectively increasing the high risk group by about 10-fold. So just to finish off a little bit, so I mean that's good as far as it goes. How could we do better? I think there are various ways in which we could improve the prediction beyond what we can do now. Most obviously, we could try and incorporate more SNPs. That's somewhat challenging because it's, the sample size is already very large. It'd be hard to get another study funded of a similar size. But we can do better by denser mapping of the known regions and trying to hone in on the variants we think are really functionally important. That should improve the risk prediction. The rare variant situation, as I've mentioned, is a bit grey at the moment. A lot of variants we don't really know whether they're associated or not. I think the risk prediction could be improved with larger targeted sequencing studies, and also whole genome and whole exome sequencing may identify new genes or new non-coding variants that may improve the contribution of the rare variants. And then on the imaging side, there are various approaches that may improve prediction due to imaging. Both there are better methods, I think, for assessing breast density, but there are also other measures, for example, parenchymal enhancement that you can measure in MRI that may also contribute significantly to the risk prediction. And then there are other potential omics strategies, methylomics, for example, which may also, there's some suggestion, may also be useful in risk prediction. Just to illustrate a couple of those points, these are, so this is CHEK2. CHEK2 is one of the known breast cancer associated genes. There's a truncating variant in CHEK2, which is very reliably established, confers a relative risk of about twofold. But there are also missense variants in CHEK2, for example, this I157T, which confer a lower risk. And so being able to get together information about all the variants in these genes, some of which we may not have detected yet, these may contribute significantly to the familial risk and the risk prediction that we haven't yet established yet. And just one final point here. So this is just looking at how one might drill down at a particular locus. So this is the estrogen receptor locus. It's a very well-known association peak for breast cancer, but when you drill down, you find that in fact it isn't one peak, it's at least 4 or 5 different peaks. And so these are each contributing independently to the breast cancer risk. And so by combining this information with the genomic information, we can hope to get better risk prediction. So I'll skip there and I'll finish here. So just to conclude, I think breast cancer risk can certainly be usefully stratified, and that potentially should improve the cost effectiveness of screening and prevention. The PRS made up from SNPs and breast density are the most informative of the available measures. And potentially the PRS can be improved by incorporating additional genomic data. I think the value of gene panel testing is currently uncertain. We need a lot more data about that. Combining all the risk factors together can be much more informative. And of course, the acceptability of stratified prevention remains to be determined. So I'll finish there. Too many people to thank individually, but I'd particularly like to thank the Breast Cancer Association Consortium, some of whose members are represented here, for all their contributions to this work, and of course the funders. So thank you.
So thank you very much, Doug, for this comprehensive survey of what's going on here right now. Are there questions?
Yes. If you tell a woman she has 40 or 50% risk for breast cancer, what should she do? What would you recommend to her? I don't know.
I don't recommend, because perhaps you should ask the person on your left there, but I'm not sure. But I mean, the guidelines, I guess, in most countries for women in that category would involve offering prophylactic surgery, MRI screening, and potentially chemo prevention with tamoxifen or raloxifene. That would be standard practice, I think.
Yeah, I guess what you mean, you're aware of the high-risk screening, but you were showing that now we can also identify women with a moderate risk by the SNP analysis. So the question is, what are we going to do there? I guess this was maybe your question.
Yeah, but I think what I want, the SNPs will also identify high-risk women. They will shift people who were previously at moderate risk into high risk, and also shift people into low risk.
Further questions? Yes, please, up there and here. Great.
So how much breast cancer genetic risk remains unexplained? And do you think that, for example, we are missing things like CNVs, which we don't really study that much, that might be contributing large amounts of risk?
Yeah, very good question. So short answer, we've explained about half. That's a very kind of global picture because it depends whether you're talking about, it's also less at young ages, it's less for ER-negative disease, it's less for non-European populations, but let's say half. I think so far the evidence is that CNVs probably, they haven't made much contribution, and to the extent they've been studied, they don't seem to be numerically very important. There are some examples, like the ApoBec blockers is a CNV, for example. So what the remaining is unexplained part. I mean, it's probably quite a lot of it, and you can show that quite a lot of it must be due to SNPs, which have too small an effect, so they haven't yet reached genome-wide significance, so that's probably quite a lot of it. And the rare variants is perhaps most of the rest, I suspect, but there could still be more of a contribution for CNVs, yeah.
One question over there, yes?
Thank you. My question is, and I don't know how it works in the UK, but when it comes to the SNPs, how many patients, or before even when it comes just to screening, have actually a SNP analysis or a gene test specific for breast cancer genes?
I mean, my guess is very low. It's not. I mean, it's not currently it's not part of standard of care. So. There's a whole question about how you would implement it. Panel testing is used to some extent in clinical genetics, so it's for higher risk women. But SNP profiling is it's really only in research settings, so there are some research studies going on, but it's certainly not a long way from being implemented generally, I think.
Okay, so then one last question over there. You might want to take this one here.
Talk, thank you. So my question was, is there any comparisons between the SNP analysis type things you've shown versus just taking a simple family history? So a simple family history, excluding the people who have BRCA mutations and stuff, which have obviously a clear picture and there's panel testing.
Yeah.
Family history can account for more common familial types of breast or even colorectal cancers and incorporate some environmental modifiers that might be in there. I'm just wondering, as a practicing clinician, I've often talked with my colleagues at Huntsman about this. A family history can take 30 seconds to do and may give you a better risk estimate than some fancy panel which accounts for, a 1.8-fold risk if you add up 30 different SNPs. What's your thought?
So, I mean, the family history is still important. That's one of the other factors to take into account, but that doesn't, the SNPs go well beyond what you can explain with family history. And remember, I mean, particularly, it's a bit different for colorectal cancer, but for breast cancer, family history is usually very limited. So most women don't have a family history. So all those women, 90% of them are all scored 0 on that. So for them, but also for women with a family history, the risk variation that you can identify with SNPs is considerable.
Thank you very much. I enjoy this lively discussion, and I think really we need to, when the SNPs are all identified, we need to look for the discriminatory value of familial history versus the genetic data. So thanks a lot, Doug.